for the invitation. I'm going to start today by presenting some of the data that's been going on at the Medical University of South Carolina that motivated my own work. Um, all with the central goal of kind of identifying the specific pathology that's associated with aging and age-related hearing loss. Quick disclosure, I have received money from Cochlear Americas, and none of that work is being presented today. For an overview, I'm going to start by defining what I see the problem as. Then we're going to look specifically at the peripheral deficits in the cochlea and the auditory nerve, and then move on to my work that focuses on the central auditory system. I'm going to try to move. I have everybody's pictures blocking my own slides. There we go. Okay. So I think the problem is we're in the middle of what many believe to be an aging epidemic, where between 2015 and 2050, the proportion of the world's population over 60 years of age will nearly double from 12% to 22%. And in fact, 2018 marked the first time in history where people over the age of 65 outnumbered children less than 5 years of age. Why is aging important to me? Here, this is meant to be a funny graphic of why aging is important, but you see these three people out on a walk. The first person says, "Windy, isn't it?" It's misinterpreted to say, "No, it's Thursday." And you see the happy gentleman thought Thursday was Thursday, and he says, "Let's go get a beer." Why does this happen? Because as we age, you can see in this demographic, we have decades along the bottom here in the x-axis, and the number of aged Americans with hearing loss drastically increases. Until the time we're in our 70s, over 50% of all older adults experience mild to moderate hearing loss. There's a big impact of hearing loss. It's the fourth leading cause of years lived with disability. And it has a lot of comorbidities, more comorbidities, including dementia and depression. It has a huge clinical impact to many of the people in the room there. There's been a substantial and steady increase in older adult presentations to ENT clinics, likely approaching 30% by 2030 for patients over the age of 65. And more than 60% of new CI candidates are all over the age of 60 years old. So if we took our nice example here, we can imagine what happens here in the clinic. And in fact, it's estimated that more than 36% of all medical errors could be accounted for due to hearing loss and miscommunication. So when an older adult walks into the clinic, their chief complaint is, "I think I have a hearing problem." You probably send them to the audiologist and we do an audiogram. I think many of you are probably familiar with the audiogram here, where we have frequency on the x-axis and hearing thresholds on the y-axis. This would be a pretty typical audiogram for an older adult. And why we'd like to think that the hearing loss in this audiogram is due or attributed to aging, we know in fact that the older adult may also have been exposed to noise, ototoxic agents. They all come with a different genetic background, as well as other comorbidities. And each one of these factors has additional factors. And yet, despite this crazy complexity that I'm just trying to demonstrate here, what do we do when somebody has a hearing loss like this? We try to fit them with a hearing aid. And yet only 20% of Americans with hearing loss actually adopt amplification. This has led to a huge kind of gap in how we can help these people. And it's not surprising that it's led to this kind of huge surge in potential biological treatments or clinical trials to improve hearing. Unfortunately, though, a lot of these drugs, as they're reaching clinical trials, have been less successful than we'd like, except for some specific genetic examples. And why that is because I think we've kind of put the cart before the horse. We're trying to treat hearing loss where we don't know the underlying pathology or the mechanisms that are actually contributing to the hearing loss. And if we're going to move from treating the symptom, which is hearing loss, to the underlying cause, we need tests that can identify the site of lesion. If we can develop these, we can actually stratify patients for clinical trials and then facilitate the selection of appropriate treatments. For example, if a drug is developed to cure hair cells or regenerate hair cells, it wouldn't help if the problem was actually related to the stria vascularis. Which gets us into the first part of our talk, where we're going to look at, can we identify the peripheral deficits that occur with age and hearing loss? The cochlea is quite complex, as I'm showing here on the left. I think you can see my mouse, where we have the hair cells shown here in purple, the stria vascularis here in yellow, and we're also going to focus here on the auditory nerve, which innervates the hair cells. The aging population is highly heterogeneous in etiology. They can have deficits here at the sensory hair cells, which would be involved in transduction or cochlear amplification. MUSC specifically has spent a lot of time looking here at the lateral wall, which, or the stria vascularis, which acts as a battery. You see this plus 90 charge here. It powers the hair cells so they can move when they receive sound input. And then we have the auditory nerve, which is the focus of a lot of my work, which innervates the cochlea and sends the signal to the rest of the system. So at MUSC, they first asked, can we use the audiogram to identify pathology? The approach here was that quiet-aged animals have different audiometric patterns than noise-exposed animals, and they wanted to determine if these patterns exist in a longitudinal database that's been collected here at MUSC since 1987. And then they're going to confirm using demographics whether those patterns were consistent with the underlying pathology. For example, if a human has an audiogram that looks like sensory loss, is it associated with prior noise exposure? We knew from animal models that the gerbil here and in human temporal bones show a decrease in the stria vascularis. So this is a microscope picture of the stria vascularis, and you can see the young control. We see a nice healthy stria, and with age, we see a reduction where it's thinning and a destruction of the barriers between the cells. Okay. I can't advance. It's like stuck. There we go. And so if you have this strial atrophy or reduced endocochlear potential, we would expect a relatively flat low frequency loss and then gently sloping high frequency loss. In contrast, I think a lot of us are familiar with sensory loss, where there's hair cell loss, which is more common in the base or those high frequencies than the apex. So it results in preserved low frequency hearing with more steeply sloping high frequency loss. So they classified all the audiograms from the database. It was, I believe, just shy of 2,000 audiograms into these 4 discrete phenotypes. And they had older normal. This is pretty self-explanatory. These are older adults who showed no hearing loss. So their hearing was normal or below 25 dB across the frequency range. We had metabolic, which was this kind of low frequency loss and gently sloping. Sensory loss, which is most common with noise exposure, where we have these low pitches that are actually better than the metabolic, but then it drops off steeply. And then we have a combination of this metabolic and sensory, which they termed metabolic and sensory. And when they looked across all the audiograms to see if this occurred, they found yes, it did. And so they had roughly 100 of our older subjects who had normal hearing. We had about 400 with metabolic, about 300 with pure sensory. And you can see the largest group here was our metabolic and sensory, where about 900 people exhibited evidence of both metabolic and sensory loss. So to kind of confirm whether these really were indicating what we saw in the animal models, they looked at how they compared to demographics. And that's what I'm showing here is that the metabolic and metabolic sensory tended to be older than the sensory or older normal. So we have years on the y-axis by phenotype on the x-axis. And you can see if you had metabolic hearing loss, you tended to be older than the other groups. And the same can be said as if you had any form of sensory loss, you tended to have a more positive history of noise exposure than whether you just had metabolic or normal hearing. So what we know from the audiogram is that quiet-aged animals and humans show similar changes, and these audiometric patterns may be consistent with demographics. More noise exposure equals sensory. So this is great. We might be able to use the audiogram. But if you looked, the majority of patients actually had that mixed phenotype, making it difficult to understand what the actual primary pathology we should be treating is. And you have to remember that the audiogram is based solely on detection thresholds, and speech is super threshold. And so far, the audiogram completely ignores anything that could be going on with the auditory nerve, which we'll get into next. So when I talk about that the audiogram is only hearing thresholds or detection, we have to know that presbycusis or age-related hearing loss is more than elevated hearing thresholds. So it may be underestimating the issue of age-related hearing problems. I like to equate it if you went in to get your vision tested and they asked you, is there a letter in this box here in the middle that the arrow is pointing to? Not to actually tell me what the letter or the word is, just can you see something in the box? But we know in reality, speech is actually super threshold and requires discrimination. And we often hear older adults say things like, I can hear you, but I can't understand you, or so am I, let's get a beer. They're not missing that you're talking, they're misunderstanding what you're saying. A lot of my work has looked to attribute this to neuropresbycusis or damage in the auditory nerve. We know that auditory nerve loss in animal models, and there's auditory nerve loss that occurs in both animals and human temporal bones. I'm showing that here in these pictures. So the top is a gerbil from our group where we showed a young gerbil, and these are spiral ganglia neurons versus the aged gerbil, which we see a large loss in the number of neurons. But the same thing is true in human temporal bones. As you went from a 2-year-old to a 61-year-old to a 9-year-old, 91-year-old, sorry, we saw a huge loss in the number of spiral ganglia neurons. More alarming is that auditory nerve loss actually exceeds all the sensory loss that occurs in the cochlea, and the audiogram is completely insensitive to auditory nerve loss. We know from animal models that you can lose up to 80% of these nerve fibers and still have normal thresholds. Luckily for us in the clinic, we do have a means to measure auditory nerve function. We can use the ABR, which is shown here on the left, as a series of waveforms, or in my lab, we tend to use here on the right, the compound action potential, or the N1. And we're looking at these peaks. The only difference between the ABR and the CAP is how the electrodes are set up, which is why it's inverted, but it tends to emphasize the auditory nerve response and the compound action potential relative to the ABR, so it's a little bit bigger and easier to see. Which you'll see why that's important in a little bit. When we get these measures of auditory nerve function, we can look at superthreshold function. Thresholds can be collected and typically follow audiometric thresholds. We don't typically do that in my lab. Instead, we look at these superthreshold measures, which can tell us about the auditory nerve integrity or function. We tend to look at the peak amplitude or how big that ABR wave 1 was, or the N1, and it's generally thought clinically that bigger is better. So MUSC, our longitudinal cohort study, has been collecting ABRs for decades. And I'm showing just the right ear from that cross-sectional data here on the right. So we have age again on the x-axis and the size of that ABR wave 1 response on the y-axis. And you can see a pretty nice progression that there's a age-related decrease in wave 1 of the ABR. We also recently conducted a meta-analysis looking not only at our data, but across all the papers that have looked at either ABR wave 1 or CAP N1 and age, and they all consistently show a robust decrease in response size with increasing age. When I say this isn't related to hearing loss, this is what I'm talking about here on the left. So I took that same data, but now we're just looking at the older adults. So this is aged 45 and up, and they've been color coded based on their audiogram. So we have normal, mild, and moderate to severe. And you can see that quite a few of our normal or the gray dots still have very small peak amplitudes, where people with some hearing loss have these big responses, which is why the audiogram is not a great tool to look at auditory nerve function. These methods can actually greatly underestimate the problem because here I'm plotting the number of people who no longer have a response present. So you can see as we increase with age, by the time we were in our 80s or late 70s, we could actually only measure an ABR wave 1 in about 50% of all participants. But this is really important for speech and communication. Here I'm looking at low probability spin sentences. There's sentences presented in background noise. By low probability, it would be a sentence like Miss White won't think about the crack, and the subject has to respond back with the word crack. Here we calculated the audibility index. So we took their hearing thresholds to estimate what we thought they should be able to hear. So we're accounting for differences in hearing thresholds. And even after we account for these differences in thresholds, we saw that larger response amplitudes, shown here on the y-axis, predicted better than predicted speech recognition. So if you had a big auditory nerve response, regardless of what your thresholds were, you tended to do better on a speech and noise task. So to summarize to this point, auditory nerve function can be assessed with the ABR or CAP. Auditory nerve function may not be well described by the audiogram, yet can help explain the speech recognition differences, particularly in older adults, because auditory nerve function deteriorates with age. But if we really want to get at characterizing auditory nerve pathology, our lab tried to take it one step further. We know that auditory nerve loss exceeds sensory loss, but synapse loss and myelin degeneration actually will precede axon loss. So that's really where we're going to want to move towards in therapeutics and intervention. Once the nerve is lost, there's a lot less that we can do. So we asked the question, can we identify auditory nerve pathology by using the ABR or that CAP? To do this, we asked what contributes to a big response? Why is it better? And that led us to develop a multi-metric approach. So what contributes to a response amplitude when we're using this clinical tool, the ABR? The first thing is the number of nerve fibers activated, which can be attributed to cochlear generators like the stria vascularis or the hair cells, the number of nerve fibers, as well as the synapses. What we were really interested in, though, is because when we're looking at the ABR, it's dependent, it's an average response, so it's dependent on the synchrony of those nerve fibers, not just the number of nerve fibers activated, which we believe, and I'll show you data, is likely attributed to the myelin status of the auditory nerve. So when I'm asking how synchrony contributes to the average response, you have to remember when we're looking at a clinical ABR, we're not looking at a single trial. We're actually usually looking at over 1,000 trials, and it's a summated response. So it can actually be a measure of about 30,000 nerve fibers, which are not homogeneous. They all respond at different levels and different time, as well as over 30 trials. So if this was a depiction of single trials in the average, and they were all the same, then our average that we're measuring in the clinic would look exactly like the single trials. In reality, it probably looks a lot more like this, where there's noise and jitter in the data, and the degree of the jitter in those responses can actually shape the waveform. So the less asynchronous these single trials are, kind of the wider the responses that we're going to see, and oftentimes the smaller the amplitude that we'll see. So my lab wanted to create a metric that could kind of capture this synchrony across trials. I had started my EEG work in the cortex, so I just stole the value from them, and it's called intertrial coherence, or we have coined it the phase locking value. And all we're doing is, instead of looking in the temporal domain, we look in the frequency domain, where we can look at the consistency of the response across trials. And that's what I'm showing here. We have time on the x-axis, frequency on the y-axis. And this bursts in color are an increase in neural synchrony across trials. So this is where the response was consistent. And we see this nice peak right where we'd see the peak in the CAP response or the ABR. The green is baseline. So we're looking at synchronous activity beyond which occurs spontaneously. But we didn't want to just look at our new value of synchrony. I believe we actually need to look across values, which is why this is called a multi-metric approach. So we have our typical peak amplitude or latency value, which has been used in the clinic for years. We also looked at something called the N1 onset, or where the response starts. And we did this so we could calculate the half width. If you remember, I said if those trials there's a lot of jitter and they don't line up, the response is likely to be wider, and we needed a way to measure this. And then we're coupling that with our measure of synchrony. And we thought that if we looked across these, we may be able to start using the ABR as a diagnostic tool. So we hypothesized if there was a loss of fibers or synapses, Um, we would see something like this, where the black line is the normal, where there's no pathology, and there's auditory nerve or synapse loss in the dotted line. So the synchrony isn't going to be really affected by this loss of fibers, but we would expect a decrease in amplitude, a decrease in width because there's less heterogeneity. And we can differentiate that between if there was a loss of myelin. So we see the same loss in peak amplitude between the loss of fibers and myelin, but now we see a huge reduction in the phase locking value. And we see that the response has gotten wider and the latency is getting pushed out. Similar to the audiogram, we turn to animal models to kind of validate this approach. So this is preliminary pilot data where we looked at auditory nerve function to differentiate these animal groups. We had two groups of animals. We had quaking knockouts, which have a myelin deficiency. And then we got data from a collaborator that used NLGN3 knockouts, which have synapse loss. Both of these groups had elevated thresholds and reduced response amplitudes, so we looked for those differences in timing. The NLGN3 data was provided to us, so we didn't have that PLV measure, but we were able to calculate that width variable. And if you remember, we said wider responses tend to have more jitter. And so we saw if we looked at the onset of the response, which was delayed relative to the width, we can pretty easily differentiate those animals that had synapse loss relative to myelin deficiency. And this is consistent with data coming out of the Lieberman lab that actually shows that synapse loss can actually reduce jitter in these animals. And we wanted to see how this looks with aging, where pathology is much more complex than you either just have myelin loss or synapse loss. And what we see here on the right is in both young mice and young humans that have had no noise exposure, the response amplitude is tightly coupled with that phase locking value. I think the correlation is like 0.9. It's actually so strong that when I originally published this data, I thought that amplitude was always related to phase locking. But it turns out if you look just in our older adults here, there is no relationship between the amplitude and the phase locking value. Here's the regression line from the younger adults, and we see pretty clearly that the phase locking value is actually better than expected relative to the amplitude in some older adults, but in many older adults, that phase locking value is actually reduced. And overall, the group, if we plotted the data, would look like here in the triangles, where we saw the reduction in amplitudes and a reduction in phase locking value. If we looked specifically at that phase locking value or a measure of neural synchrony, we did see that that is also associated with poor speech recognition. So the top graph is showing time compressed speech, where we took, I believe they were spin sentences, low probability, and we increased the rate. And we saw that if you had poor neural synchrony, you were less able to repeat those words. And the same thing if we looked at speech and noise, and this is the Quickson data. But does weaker synchrony actually is it associated with poor myelin integrity? To look at this, we try to put our subjects in the scanner to look at imaging of the auditory nerve. We use DTI. DTI is typically used to assess white matter and fiber tracts in the brain. And I'm going to show data from fractional anisotropy, or FA, where higher values are generally associated with healthier myelin. We're also measuring something called the quantitative T1 relaxation, where shorter relaxation tends to equal healthier myelin. There's some evidence if you use multiple metrics and they both tell you the same thing, it's more likely that that's actually what's going on. So that's why we use two different measures of myelin. This has been used in other studies to look at peripheral nerves, which I'm showing diffusion tensor imaging of the arm over in that image on the right. And it can track the status of axonal tracts during damage and repair. And its specificity to myelin, though, is currently unknown. So here's what the imaging looks like. I've inverted the quantitative T1 image on the left. So if you're used to seeing these, you're probably used to the nerves being black voids. All I've done is inverted the image because I think it's easier to see the auditory nerve if it's white. And so that is the line here. We have the cochlea at the top and the semicircular canals. And you can see here the vestibular branch of the cochlear nerve. Over here, a little bit less clear, but this is our diffusion weighted image. This is the fractional anisotropy image, and this is our auditory nerve leading to our cochlea and our semicircular canals. And so when we look at that data, I'm plotting now the FA, but the QT1 would show the same thing. We saw a decrease in FA with increasing age in our older adults. And it was very tightly related to that measure of neural synchrony from the auditory nerve. So increasing PLV or neural synchrony was associated with better fractional anisotropy or better myelin. But these measures still aren't necessarily specific to myelin. So what we've recently started and hoping to get funding for is to look at this in temporal bones where we can image the temporal bones, but then also confirm it with pathology. So here's one of our temporal bone specimens that we have opened up so you can see the cochlear nerve, crudely circled here in red. We put it in a tube and we can actually image the auditory nerve. And this is our high resolution image. And then this is that same sort of FA image where we're looking more specifically at myelin. And again, you can see the auditory nerve running in the internal auditory canal and the cochlea. This is just another picture of the histology. My collaborator, Hainan Long, has developed this nice approach where she can actually just use a hammer and open up the auditory nerve to expose the cochlear nerve and visualize the cochlear nerve and the facial nerve. And the important part here is that we can do the histology on the same bones we're imaging. And so this is in one of our temporal bones from a 57-year-old versus an 89-year-old, where the green here is what we're looking at, and that is the myelin stain. And we can compare that to the same diffusion metrics we're getting in the scanner. So when we looked at, we've scanned 3 bones so far from what we're calling a young old bone, which was 57 years old, as well as 2 from older donors who were 88 and 93. And we saw that the diffusion metrics were consistent with what we saw in the in vivo humans, where FA decreased with increasing age, and MD does the opposite. That's what happens with the diffusion metrics. But importantly, we can then look at that relative to the things like the G ratio, where we can plot out myelin thickness, where again, we saw that here we had 2 bones with histology that were younger, a 57 and a 59-year-old. And we saw that the myelin thickness was higher in those bones relative to our 2 older old donors. Hainan has worked out a new method of cochlear clearance, which I'm showing here to give myself a break too, but to show you that the structure of the auditory nerve is heavily myelinated. That's what is shown in green here. And our approach here is we're going to use Amaris to actually calculate myelin across the entire structure so that it's better comparison to when we're doing in vivo imaging in humans. So to summarize up to this part, there's more to the ABR than amplitudes or thresholds. Examining differences across these measures may be key to determining pathology. DTI structure histology association suggests age group differences in myelin integrity of the auditory nerve with increasing age. And in the future, we're trying to use machine learning to classify the actual pathology that's present in both the auditory nerve and the cochlea. And this is data from a paper that I worked on with Karen Steele in the UK, where we weren't using it to look at myelin specifically, but it turns out we can use these same techniques to identify strial deficits. When the strial is damaged, we tend to see that the phase locking value is relatively preserved compared to the amplitude. And you see that, and you can look for more information in this paper. The strial animals looked more like the healthy controls when we looked at the PLV relative to amplitude compared to our sensory loss animals. Okay, and so now we're at the final part of the talk where we're going to move on to the central auditory system, specifically looking at deficits in GABA and myelin. I like to say this is because we hear with our brain, not our ears. And I think in our field, we tend to just look at the ear and ignore what goes on beyond that. But this is actually especially important in aging. We know, again, that presbycusis is more than elevated thresholds, and there's actually age-related changes throughout the auditory system. that are both sensory driven and age related. I'm showing here the left side of the brain is a healthy young person. The right side of the brain is supposed to represent an older individual. We can see ventricle enlargement, cortical thinning here of the gray matter, tremendous volume loss, sulcal widening, just a lot of damage. This is data from a longitudinal study here at MUSC, where we looked at how age and hearing loss contribute to cortical atrophy. So during the same time period, which areas of the brain had atrophy relative to hearing loss? But I don't want to just continue to look at atrophy. Similar to when I'm looking at the cochlea and the auditory nerve, I want to look at specific areas where we might be able to intercede for intervention. So we tried to identify neural factors that may relate specifically to speech recognition, are altered with hearing loss, and amenable to treatment. So we're going to look at GABA and myelin for the rest of this talk. Why GABA and myelin? They are both key players in neural degeneration and function and plasticity. They're crucial in maintaining temporal precision, which is important for speech. They may be potential targets for intervention. There's actually already pharmaceuticals available to modulate both. And luckily, through imaging, they can both be measured non-invasively. So the first part, we're going to look at a paper that we published, I guess, several years ago now in 2022, where we looked at how changes in the auditory nerve contribute to changes in cortical GABA and contribute to what we called central gain in older adults. I will say why this study was the first time to show this in humans. Most of my work just takes what smarter people than me did in animals, and I try to do it in humans. So this is already well established in the animal world. But we wanted to look at how the aging brain reacts to the loss of input that I just spent the first 30 minutes telling you. And when we're talking about GABA, we're talking about inhibition and the balance between excitation, which is generally associated with the glutamate as a neurotransmitter, where when we're talking about inhibition, the neurotransmitter is GABA. And when we have hearing loss, there seems to be this shift where we have a decrease in inhibition relative to excitation. As a hearing person, I like to think of this as the volume wheel. The brain likes to maintain a set point, and when we saw that decrease, the brain responds by decreasing inhibition or turning up the volume. This is not unique to hearing and the auditory system. This is an example of homeostasis, where the body just wants to maintain internal stability. An example is that you shake when you're cold or you sweat when you're hot, all to maintain this kind of level in the body. But how does this look in the auditory system and how do we measure it? Well, with sound. Through the ear, we typically have those spiral ganglion neurons that lead to the cortex. And in general, we see a bigger response in the auditory nerve, we expect a bigger response in the cortex. So what happens in the aged auditory system? Hopefully you believed that there's this decrease in auditory input due to this loss of spiral ganglion neurons, which you would think should contribute to this decrease in the central auditory system. But the brain doesn't like that, so we have that volume wheel, and this is what we call central gain, where we actually see an increase or a maintained response in the cortex of the older adults relative to the input received. We can measure this sensory loss with our CAP or ABR. We can measure that decrease in inhibition or GABA in the scanner, as I'll show you. And we can look at this cortical response by looking at instead of the auditory nerve response, the cortical response. So here we have a P1, an N1, and a P2. So these are just cortical responses that occur a couple hundred milliseconds after sound onset. And then we wanted to see how this system relates to function. Is bigger still better, or is it actually maladaptive and worse? So I've already shown you that auditory nerve responses are reduced in older adults relative to younger adults. This is just the data from that specific paper where we saw this huge reduction. The red is the younger, the black is the older adults, and the older adults had significantly smaller N1 responses. Then we put our older adults in these caps. You could see how happy it makes them not to be having the TM electrode anymore. And they get these huge responses. So The same adults that had no auditory nerve response now have this huge cortical response, where we see this enhanced P1, where the response is actually significantly larger in the older adults relative to the younger adults, and a similar size response at the N1. If I show you the individual data points for all the younger and the older subjects, this is from the auditory nerve and this is from the cortex. And we saw a significant decrease, as I've said, in the auditory nerve response, and it's missing its bar here, but we saw a significant increase in the cortical response. So despite this really small input to the brain adapts and we see this much larger or preserved cortical response. So in our younger adults, we found that the auditory nerve response predicted the cortical response, which is shown here. So the CAP amplitude relative to the cortical amplitude. So that if you were a big responder at the auditory nerve, you tended to be a big responder at the cortex. When we looked at the older adults, we found that that wasn't true. So to calculate gain, we wanted to use that association in the younger adults. So we took this regression line and then we said in the older adults, if you acted like a younger adult and we had your auditory nerve response, what would your predicted cortical response be? It's similar to how we looked at that predicted speech recognition based on the audiogram, but it's just using a regression prediction approach. And what we found, if you were on this red line, then your observed response looked like your predicted response. But in the great majority of older adults, the response in the cortex was actually much larger than what we would have predicted based on that auditory nerve response. And that bar graph is shown here, where the observed cortical response is significantly larger than predicted. And we wanted to know, was that related to GABA? To do that, we put people back in the scanner. We used a sequence called Megapress, which is a fun name, but it's honestly just measuring GABA in the cortex. We measured GABA from these two purple squares. This one is on auditory cortex. This is just a control region, non-auditory. And what we found was that less GABA, so a decrease in inhibition in the auditory region, was related to more central gain. So this is very similar again to what they found in animals, that when there's a decrease in GABA and inhibition, we see this increase in central gain. And I'll say this was specific to the auditory region. In our control region, this relationship was not true. So it's not likely a general aging phenomenon, but instead specific to this loss of afferent input. So is bigger better? Did the brain do this because it helps us? Unfortunately, no. Central gain is actually associated with poor speech recognition and noise. So now we're looking at that central gain mechanism on the y-axis. So more central gain is this way. Sorry, I don't have an arrow. And that was related to poor speech and noise recognition. And this was for the Quicksand data. So to summarize on central gain, reduced afferent input from the auditory nerve contributes to decreased inhibition measured with GABA, cortical hyperexcitability, and poor speech recognition and noise. Now to the final part of my talk. Good. If you're still with me, we're going to look at myelin deficits in the central auditory system. Myelin and aging. Myelin becomes thinner, shorter, fragmented. We know that there's reduced repair by oligodendrocyte precursor cells, but repair is still possible. I think it was the dogma for a long time that we kind of gained myelin through development and then it became stable and didn't change. We know in the last decade that is not true at all, that myelin is adaptive and continuously changing and repairing. They've shown imaging studies where you can teach people to juggle in 10 weeks and actually show changes in the brain associated with myelin, even in older adults. We know in mice, if we we knock out these oligodendrocyte precursor cells, older mice can't learn a task anymore. So myelin is very plastic and responsible for changes all throughout aging. They're a core feature of brain aging, directly tied to everything you could ever think of, but not often considered in relation to hearing. And they can be measured non-invasively with MRI. So we know the auditory system is highly myelinated. I showed you a pretty picture from the auditory nerve, but we know that all through the brainstem and cortex are also highly myelinated. Myelin is crucial for efficient neural transmission, which is needed for speech recognition, and it's adaptive and dynamic. I showed you we are looking specifically at myelin in the auditory nerve, but we can also do it at the cortex. So if we're talking about diffusion tensor imaging, we can actually pull out all the white matter fiber tracts in the brain, or just the white matter in general, which we're showing here. And when I'm talking about diffusion tensor, we have some general patterns that we look for that are associated with white matter integrity. Generally, higher fractional anisotropy is good, low radial diffusivity is good, and low mean diffusivity is good. So we reanalyzed data that we had been collecting through the years that we haven't published yet. And this is looking at radial diffusivity, but I could have shown the opposite relationship if I looked at fractional anisotropy. But in this graph, poor myelin integrity is up by age. And this was just a large group of older adults that we had diffusion tensor imaging on. And then I looked at those with normal hearing and those with hearing loss. That's the color coding. And then they were pulled out here. And we showed both a significant effect of age, that myelin integrity decreased with increasing age, but it seems like hearing loss actually compounds the effects of age and makes auditory white matter integrity and auditory cortex poorer with hearing loss than with normal hearing. In a paper that we have under review, we showed again that this white matter integrity is actually important for speech recognition. So this is plotting fractional anisotropy. So now high values are good. I apologize for that. And high FA was associated with better word recognition. So the diffusion data that I just showed you is sensitive to myelin, and it establishes that myelin degeneration may occur with age and hearing loss, but unfortunately, diffusion imaging is not specific to myelin. Alternatively, we have new imaging tools that we're working on that are specific to myelin, including the VISTA and the QT1, which I showed you for the auditory nerve. And again, we want to look at whether we can look at converging evidence across these measures to increase our confidence. So this is an example of VISTA, which is a myelin water fraction imaging. So all the white here you're seeing, or light gray, is myelin. This sort of sequence looks at the water trapped between lipid layers to do some crazy calculations, which don't ask me about, to calculate what the myelin water layer is between those values. And we looked at these myelin water fractions. So higher myelin water fraction means more myelin. And we looked at it relative to our EEG synchrony and latency measures. Oops. And this is our preliminary data. So we looked at the myelin water fraction from the inferior colliculus, and we saw an incredibly strong relationship to our wave 5 of the ABR. Better synchrony was associated with more myelin, and this was enhanced when we increased the click rate. So we went from a slow rate to a fast rate, which put stress on the myelin, and it actually made the relationship stronger. We saw the same thing if we looked at auditory cortex myelin water fracture in relationship to a fast rate click and latency values. So more myelin, faster latencies. And the same thing was true when we looked at a speech syllable and noise. And these are data all from older adults. To summarize, both age and hearing loss contribute to neuroplastic changes in the central auditory system, including GABA loss and myelin degeneration. These deficits contribute to poor speech recognition and noise, and they may be amenable to intervention. I'll thank everyone who has helped me.